Hello everyone, my name is Jahid Mia and I'm a biology and biochemistry double major here at SUNY New Paltz, and this summer I did research with Dr. McBrayer evaluating quorum sensing reporter assay conditions. And what is quorum sensing? Quorum sensing is a population density dependent communication pathway used by bacteria to activate and coordinate group behaviors such as biofilm formation. Bacteria are able to monitor their surrounding population densities by continuously secreting signaling molecules into the extracellular environment. And once a certain threshold is met, these quorum sensing circuits will activate. And these circuits can be activated in two ways. Either the receptors that bind to the signaling molecules act directly as a response regulator, which increases the expression of the quorum sensing genes, or these receptors can initiate a signaling cascade, which ultimately results in the upregulation of these quorum sensing genes. In gram-positive bacteria, such as B. subtilis, which is used in this experiment, use peptides called auto-inducing peptides as their signaling molecules. These peptides have to be modified and exported out of the cell and re-imported back into adjacent cells to activate the quorum sensing circuits. And utilizing a reporter strain with the plasmid encoding for beta-galactosidase under the control of the COMEX promoter will allow us to conduct bioassays to investigate the crosstalk between closely related bacillus species. And finding the optimal conditions for these procedures will allow us to increase the sensitivity and accuracy of our results. And the research questions we have this summer were: what are the optimal conditions for our reporter assay, and is there crosstalk between closely related bacillus species? And the major breakthrough from our research was that we were able to optimize the growth conditions of our bacillus species. And we were able to do this primarily through the use of an Erlenmeyer flask to properly ensure the oxygenation of our medium. We could see here that bacteria grown in static conditions such as a microplate or a test tube really struggled to reach the growth observed in bacteria that are grown under our optimized conditions, such as in the Erlenmeyer flask. And B. megatherium was a species that only grew using our optimized methods. Here we see here that in the plate growth, the bacteria did not grow at all, but when under our optimized conditions, the bacteria really flourished. But our optimization efforts for our bioassay has failed to produce a significant signal above the background noise. In our bioassay, bacteria that were dead produced a higher signal than bacteria that were alive. And we knew the bacteria was dead by monitoring the growth during our bioassay, and a growth curves using the highly concentrated licheniformis supernatant confirmed that this supernatant has a cytostatic or cytotoxic effect towards bacteria. And in the future, we will use a dilution series to confirm whether the supernatant is cytostatic or it is cytotoxic. And in the future, we'll have to use a chemically defined media that is used in the scientific literature when conducting our bioassay. Normally, bacteria are able to express their quorum sensing circuits in normal growth conditions, but we suspect that B. subtilis requires stressful conditions to chorate. And I would just like to thank a few people. I'd like to thank Brianna for coming through during the summer and helping us out in the lab and being generally pleasant company, and Melina for volunteering during the summer to help us out during the lab, and the RSCA for funding this research and making this possible, and you, the viewer, for checking out my presentation. Thank you so much, and have a wonderful day.